Namaste to all. Welcome to the new session on drug discovery and drug designing. So in this, we are going to see about the drug target identification and validation. So in our previous session, we have seen that how to use different tools, or you can say the methods for drug targeting. And in this particular session, what we are going to see is we are going to see the genetic applications of drug designing or drug targeting by identifying the correct loci. So this is called as gene tracking. This particular whole session is called as gene tracking, or you can also say this as mapping disease loci. So this whole session, I'm going to divide this in two sessions. So in today's session, we will have an introductory part. So in this, you will be learning the basics about the mapping part and how the disease is being there or how the loci is being there, what is loci, everything you will see in this particular session. In the next session, we will see about the gene tracking. So in this mapping, the basic idea is starting with our central dogma of molecular biology. And in the central dogma of molecular biology, the important aspect is our DNA. So the DNA is acting like the basis of life and disease. We know that each DNA is having many codes in the form of letters like adenine, guanine, cytosine, and thymine, which are being bounded by the bonds, like hydrogen bonds are there. So adenine pairs with thymine and guanine pairs with cytosine. And the hydrogen bond between the adenine and thymine is double bonds, and guanine and cytosine is triple bonds. So based on this, if any mutation happens or based on that, there is a particular sequence which can be for a particular disease also. So that when it gets highlighted during the whole cycle, that is when it is getting expressed and the protein translation takes place, the proteins which are being releasing this are intended to increase the particular symptoms for the particular kind of disease or disorders. So this can happen in any time in the lifetime. So that is what is actually about the whole thing. So in this whole process, what we require, we have to understand about the DNA sequence. So when we see DNA is the deoxyribonucleotide. So in that, if you see the sequences part, in that, what we have to see is we have to see about the nucleoside and nucleotide. Nucleoside, if you see, this is called as base with your pentose sugar. This is called as your nucleoside. When this nucleoside, they join together with another nucleoside with a phosphate diester bond, phosphodiester bond, then what we have is we get our nucleotide. So the composed form of three parts, that is a base, pentose sugar, and the phosphate group, it tends to form the nucleotide. And when the nucleotides are all present, then what happens is we actually get to have our sequence. That is, because of the phosphodiester formation, one nucleotide joins with another nucleotide by the 5' to 3' direction or 3' to 5' direction. And when they are being joined by the hydrogen bonds, as I said, the double bond or triple bond, like in between the adenine and thymine and guanine and cytosine, they tend to form the DNA structure. When this becomes elongated form of many things, they tend to form the sequence. So a DNA sequence or the genetic sequence is a succession of letters representing the primary structure of a real or hypothetical DNA molecule or strand with the capacity to carry the information as described by the central dogma of molecular biology. And as I said, the letters which are present here, which are representing the adenine, guanine, cytosine, and thymine are A, C, G, and T. So what happens next? You know that when the DNA, that is nucleotide, is there, you will have your histone molecules around which the DNA gets wrapped around. And when the solenoid model, when they compress together, they tend to form the chromosome. So in this particular chromosome, you can see that there are so many places are there where it has been marked that in the future, what kind of particular disease or syndromes or the particular problem or disorders can happen. Like ocular albinism, then chronic granulomatous disease, then Duchenne muscular dystrophy, Meckel syndrome, testicular feminization, then X-linked severe combined immunodeficiency, then you are having your X-linked gamma globulin anemia, then Fabry disease, then Leishmanian syndrome, then you are having hemophilia B, Hunter syndrome, Fragile X syndrome, hemophilia A, then G6PD deficiency. So these are the different kinds of disease or syndromes or disorders which can occur in a particular chromosome. So here, how we have to find it out or how this is happening. So here, if you see what is locus, that plays a very important role. So locus means a unique chromosomal location defining the position of an individual gene or DNA sequence. Like, for example, this is our particular position in which a particular DNA strand is there or sequence is there. Then in that particular sequence, what are the positions are there or what are the different genes are there or the sequence there that is actually representing the locus or loci. Loci means if it is in a plural form, so it is there in different places. Locus means a singular form, so it is only present in a one chromosome or one arm of the chromosome. So basic definitions, if you see in this particular part, is allele. So what we consider with the allele is one of the several alternative forms of the genes or DNA sequences at a specific chromosomal location. This is called as locus. At each autosomal location, an individual possesses two alleles, one inherited from the father and one from the mother. So as per the Mendel's genetics, you know that one is coming from the mother, one is coming from the father. When they join together, they are going to form the zygote. And in that it will be, that is haploid to diploid formation will take place. Then the phenotype, if you see, that is the observable characteristics of a cell or organism, including the result of any test that is not a direct test of the genotype. So what you can see from outside, what are the different features, what you see from outside that plays as a phenotype, and what you see from inside, that is basically the genetic constitution of an individual, either overall or at a specific locus, is actually called as the genotype. So in this, if you see, what is meant by heterozygous? Heterozygous means an individual is heterozygous at a locus if he or she has two different alleles at that locus. That means if it is coming from one from the mother, one from the father, then it is a heterozygous. If it is in the same, that is the fathers only, if you see that the mitotic division has occurred and it has formed, then it is a homozygous. That is, an individual is homozygous at the locus if he or she, that is, has two identical alleles at that locus, then it is homozygous. That is, if two different places are there, like for example, this is the arm which is present. If one is present at this locus and one is present at this locus, this is heterozygous. If both are present in the same part, then it is homozygous. Then haplotype. Haplotype means a series of alleles found at linked loci on a single chromosome. That is, in a single chromosome, if it is linked, that is, if it is joined nearby, we can say that is a haplotype. So what is meant by primer? If you see primer, that is a short oligonucleotide, often 15 to 25 bases long it is. So as we have seen earlier also for molecular biology, that a primer is acting like a starting position, that is the starting sequence. So it has to be of a lesser length. So this primer is an artificial primer, or you can say if it is taking place inside our body or in a cell, that is a natural primer. So natural primer, in some cases, you can say that this is the RNA primer. So in the normal case, when we are doing the PCR or the DNA sequencing, for that we require the primer. So this primer is, as I said, is an oligonucleotide sequence of 15 to 25 base long, and the pair specifically to a target sequence to allow the polymerases to initiate the synthesis of the complementary strand. So as we know that if it's a DNA strand is present and I want to make a copies of this particular DNA strand, if I add my primer, one will attach at the 5 prime to 3 prime, another as attaching at the 3 prime to 5 prime, and the movement will be in the 5 prime direction, 5 prime to 3 prime direction only. So what happens here is you will start to have the new strand formation, and you can have multiple copies of the same DNA. So mutation change the structure and function of DNA. So how it occurs? If you see, we have seen different kinds of mutation in our previous session of molecular biology and RNA technology. So here, if you see, like if you are having a sequence, and in this particular sequence, instead of you are having the tryptophan, that is the TGG, instead of that, it has become as TGA, that becomes as your stop codon. So this is actually called as substitution mutation, or you can that also called as nonsense mutation. The reason why it is called as nonsense mutation is by the conversion of a small base at the third position of the codon, what is happening is because of the substitution of this base, this is called as substitution mutation. In other words, when we are changing the sequence, instead of tryptophan, it is becoming as stop codon or nonsense codon. That is why it is also called as nonsense mutation here. Then other one is called as missense mutation. In the same format of the substitution mutation, in place of the guanine, if we are having the cytosine, this is going to give us a next sequence. Instead of tryptophan, it is going to have the cysteine, and this is actually giving a missense. So we cannot say that this is going to give a correct sequence, but in that sequence, the amino acid, since that changed, the property can go wrong also. And frame shift mutation, that is instead of reading in the same either by the deletion or by the insertion, they can have a change. So if you see in this particular example, that is the deletion of the C shifts reading the frame, creating new amino acid sequencing. So if you see, that is in place of C, it is being changed. So what happens? The GAA, ACC, then you are having AAT, and then you are having GG. So because of that, what is forming? You are having, instead of first one is alanine, the second is glutamine, threonine, and last one is aspartame. So what happens, aspartame? So what happens here is because of a single base change, the whole sequence of the amino acid has got different. So the next part in this is the deletion. So in this deletion, if you see one of the base, if it gets deleted, the size of the particular strand actually decreases, and it can lead to various kinds of problems. And the same way, the duplication, that is the same base, if it is getting duplicated, if a base type is there, like GTT is there, that is again duplicating, the size of the particular strand will increase, and due to the increase in the strand, there is a chance that the persons can have some kind of deformities in their growth while it is taking place in the womb or outside the body also. So insertion mutation, in the place in between, if you are adding one more, the size may differ. Also, the properties can be differ also, because there can be a new property or new amino acid can be synthesized, and due to that also the changes can occur. Expansion, if you see, the expansion is like a combination of this itself, like a duplication at multiple times. So what happens? It becomes a very huge sequence. So since the length has increased, there can be disease or there can be some deformities or some changes. Then inversion, so in place of TG, it has become GT. So the property actually changes here. So like this, there are so many things are there which are associated with this disease mapping. So introduction to this is the differences in the DNA sequences between any two individuals. It is nearly like 0.1%. So the sequence variant version or alteration, if you see, it can occur at any sequence change can occur in this. Also, the important part in this particular mapping disease loci is polymorphism. Polymorphism means if a sequence alteration is present in at least one population, 1% of the population, then this is called as polymorphism. Polymorphism means poly means many, morphism means the structure change, or it can be of the particular position or phenotypic change, anything can be. So the example of the intergenic polymorphic sequences like SNP, SSR, or RFLP, etc. is present. So SNP means single nucleotide polymorphism and SSR means simple sequence repeats. So these changes actually occurs in the DNA during the process. So what is meant by single nucleotide polymorphism? The most common sequence variations are single base changes, that is called as SNP. So in this, what is happening is that is less than 1% of SNPs occur in the coding regions and alter the gene product, and this leads to the disease. So you can see that in the marker that is co-inherited with a disease causing gene due to the physical proximity. So this particular gene which is present, like that is the SNP which is present, they also act as a gene marker. So what happens is we can identify that at this particular loci, our particular disease gene is present. See, for example, if you are having a sequence in this, like GC is present, in this, the upper strand is 160 unit and the bottom strand is 150 unit. So in this, if the changes has occurred, that is, it is known that the person experiencing the side effects often have more G, that is the guanine bases in their gene. So due to this, what is happening is there can be either reduce or there can be an increase in this particular sequence, and then due to this particular changes, what happens is either there can be the length can be decreased, or there can be a huge growth, or their brain will not be developing at some times. So many changes can occur because of the particular gene change or in the particular loci problem. So the simple sequence repeat SSR, that is the critical markers in the genetic study linkage studies and in forensics or medical identity testings. Then highly pleomorphic between two individuals. Then on average, one SSR occurs every 2000 bases. That is the fundamental of the molecular diagnostics. So in SSR, there are two types are there. The first type is called as microcyclides or short tandem repeats, that is STRs. So the short segments of the DNA, that is the 2 to 6 base pair long, that are repeated end to end, and mini satellites, if you see, that is the variable number of the tandem repeats, that is the VNTRs. So the repeated segments of the DNA that are 15 to 70 base pair long. So that is present here. So what is the DNA marker here? So if you see the DNA marker, that is the polymorphic locus on the gene that is easily assayed, yielding reproducible result. So that is the main importance of the marker. So marker, what we are having here is we are at different locations. Okay, so polymorphic locus means like 100 base pair, 200 base pair, 300. We can see in this particular, there are different ranges are there, like a ladder. This is looking like a ladder. Okay, so in this, what is happening is we are having 100, 200, 300, 400, 500, 600, like that till 2000 base pairs. So this actually gives us an idea, and when we run our sample, we can identify that at which position this particular locus is present, and that is the gene is present, and we can identify easily that this particular disease is present at this particular locus, means this particular patient is also having the same symptoms or not. Then use of the genetic or the DNA markers, if you see, to identify the chromosomal location of the mutant genes associated with hereditary disease. Then the second one in this is the DNA fingerprinting and individual identification, then determination of the relatedness and identify in transplantation, then cancer genetics, then paternity testing, then detection and quantification of transplant chimerisms in allogenic BM transplant, that is the bone marrow transplants, then epidemiology and food safety sciences, then human population history and anthropology, that is improvement of domesticated plants and animals, then ecological indicators, evolutionary genetics. So these are the different things what we can understand and use for using the genetic markers. So regarding the mapping part, the first thing what we have to understand is, as per the according to the Mendel's third law, which states that the alleles of the gene at the different loci segregates independently. Based on this is true that genes on different chromosomes will have different loci. So this actually gave or proposed for the genetic linkage studies. So this says that if two loci are positioned close together on the same chromosome, so that the alleles at these loci are inherited together, more often than not, these loci are said to be linked. That means in a same chromosome, if the two loci are nearby, they are linked. If it is far away, they are on the different loci. So recombination linkage, if you see here, that is a recombination fraction that is given by the symbol theta. So this is the measure of the distance separating the two loci. See, for example, one loci is at some around like 10, another is at around like some 20. So the distance between the two, if you see, that is the 10 loci. So that is how the determination is being done. Then centimorgan, that is the cm, small c, capital M, it is called as the map unit, and the unit of measurement for genetic linkages. And if two loci are 1 centimorgan apart, recombination frequency is 1 in every 100 meiosis, that is theta is equivalent to 0.01. So greater the theta value, greater is the chance of recombination and lesser is the chance of being linked. So you understand that if it is far away, if it is far away, that means the chances of linking is very less, but the recombination will take place. So if two loci are not linked, then the theta is 0.5 and above, you can say. So linkage analysis, if you are going to do, that the basic methodology involves the study of the segregation of the disease in large families with polymorphic markers from each chromosome. So eventually a marker will be identified so that the co-segregates with the disease more often than would be expected by chance. That is, the marker and the disease loci are linked. So if you see in this particular picture, the male is infected, female is not, but the children here who are born to them, they are all maximum, they are carriers who are not infected, but if they are infected, they are almost getting affected, like female and male both are affecting here. So if you see the total number here is 8, in 8 you are having like 3 are getting affected, and it is being shown here, the disease is shown. So if they get married, what will happen? If a normal child is getting married, the girl is actually acting like a carrier here. So what is happening is, when they go to the new family, when they have their progeny, what will happen is, out of 5, one will have the particular disease. In the same manner, when one of the normal child is again getting married, in them, out of 4 one will have the disease. So this ratio, what you can find from this is, one set is, it is 1:4 or 1:3 category, you can say. And another case, if you see here, this particular case, in this also the same scenario is forming, but here from female, it is when it is getting transferred, the affected one is male only. So here what is happening, it is also like 1:3, but once it is getting transferred from female, then what happened is that males who are forming, that is like 2:2 ratio. So 2 are affected and 2 are not affected. So this is how the linkage analysis is being taken place, that you can find a genetic analysis, genetic studies you can do from this particular experiments using the mapping of the particular or the disease loci. So the genetic linkage, if you see, each DNA polymorphism serves as a genetic marker of its own location in the chromosome. And in the pedigree, if you see whatever we have seen, this is called as pedigree. So the importance of the genetic linkage is that the DNA markers that are sufficiently close to the disease gene will tend to be inherited together with the disease gene in the pedigrees. So if you see the DNA polymorphism, where all it is getting affected, you can see here. So in this particular, this is a disease gene which is present in the Q region. The upper region of the chromosome is called as P region, and the bottom part of the chromosome is called as the Q region. So the Q region is always bigger. So in the Q region, if the D is present, that is the disease part is present, then what happens? The nearby gene, what are there? They are actually the close enough to the disease gene. So genetically linked. They are genetically close by, so they are genetically linked and inherited together. Inherited together means when they are getting transferred to the next generation, that also comes, that particular gene also comes along with the disease gene. The second part in this is, if you see the last gene in the Q chromosome, that is the Q part of the chromosome, that is too far from the disease. So it is far away. Okay. So the distance between them is far. So you can say that when the distance is far, the greater the distance, lesser the chance of the disease, you can say. So what happened? Not linked. So not inherited in the pedigree. So that is why what happens is, in the next generation, when they are going, they are not expressing any kind of disease in this. If you see in the upper part, that is the P part, what is happening is the DNA polymorphism along with the chromosome. That means it will also pass on with the next generation. So it is also going to the next generation. So in this particular case, what happens is, when the transfer takes place, when the mitosis or the meiotic division takes place, they also get transferred along with this particular chromosome. So this is what is actually the DNA polymorphism actually teaches us. So like this, when we study for many genes, then this is called as genetic mapping. So the initial approach to the identification of the disease gene is to find DNA markers that are genetically linked with the disease gene in the order to identify its chromosomal location. It is called as genetic mapping. So if you see the linkage studies, what is linkage studies? A method using the DNA markers physically adjacent to a disease gene, that is linked gene. Then what we do is, in this what we study, this indirect analysis allows the disease gene to be tracked through a family. So as we have seen in the previous, so if I am going to study, if I find that near to the disease, the whole pattern, if the near to the disease gene are always present, that means the disease gene is also present. So once we understand that the disease gene is also present, from the linked gene, we can identify that, okay, the nearby, it can be a indirect analysis. This is called as indirect analysis, which shows us that the disease gene is also present. So this way, the genetic status of at-risk individuals can be determined even when the identity of the disease causing the DNA sequence variant is unknown. So still, if you don't know that this is going to cause any disease, based on your study, based on your sequencing, you can identify that whether the disease will occur in future by the linked gene which are nearby. So the molecular basis of the inherited diseases, if you see, there are two strategies to isolate and characterize involved genes. That is, first one is the functional cloning or classical approach or classic approach. That is to study the variety of inborn errors like PKU or disorders of the hemoglobin synthesis like anemia and other things. You can see this particular thing, or the knowledge of the abnormal gene product and the corresponding affected protein is known. So usually the practice, still now the practice in the pediatric division of the medical sciences is, if a newborn baby is there, after about a week, what they do is from the palm region, that is from the feet region, that is what you can say like the foot region, they actually puncture and they take the blood samples at different papers are there. So in that they take the blood samples and they send it for the analysis. That is neonatal analysis it is called as. So what happens is when we send for the neonatal analysis, we can know that whether this particular baby is having any kind of problem or not, or whether any disease genes are present in them or not. That report analysis, we can do it and we can understand. So the positional cloning, or the candidate gene approach, this is called as. In this, what we are understanding is no clue to the nature of the defective gene product. So we don't know from the normal thing that there is any defective genes are present, but relies on the mapping the disease phenotype to a particular chromosome location, and accomplished in the disease is associated with the distinctive cytogenetic change or a linkage analysis. So what happens is in the cell development after about like 7th month or 6th month, people actually they take the samples, neonatal samples like amniotic samples, and they test for the particular disease, whether they are having any genetic disease or anything like that, and they check it. And it is very useful for many of the babies. Before giving birth itself, we can know that this disease are there or not, like the cystic fibrosis, NF, DMD, PKDs, or Huntington disease. So like this different disease we can identify. So cystic fibrosis is a hereditary disorder characterized by the lung congestion and infection and malabsorption of nutrients by the pancreas. So what happens by the cystic fibrosis? People will have the autoimmune problems, or related to that, they will have the metabolic problems, or they will have the respiratory failures or respiratory problems or heart problems. And in future, what happens is they cannot do any athletic works or anything like that, and they can have the different kinds of scenarios. So in this session, it is like you can say like 50% of the sessions only you are having right now. It is the basics for understanding the gene tracking. So in the next session, what we are going to see is we are going to see this particular topic, that is how the different gene mapping is being done for the loci and for understanding the disease aspect, that is called as gene tracking. So I hope that you have got an idea about the gene tracking or the base of the mapping disease loci. So see you in the next session about the gene tracking, and have a good day. Bye. Thank you.